Hi everyone, my name is Beth Neace and I'm an instructor in the science department of WTMA. I currently teach biology and anatomy and physiology. I am a military wife, which has not only allowed me to live in many different places, but also to teach at various levels of education. I have taught 8th grade science as well as high school science in the form of biology, anatomy and physiology, and chemistry. My love of science started when I was very young. I had a 5th grade teacher that really sparked my science curiosity, and it stayed with me for the rest of my life. It is my goal for every student that I teach to find something in biology or anatomy and physiology that really sparks that curiosity and makes them ask the question, why? It'll also kind of show them that there are worlds that they didn't even know exist, and who knows where that passion and that curiosity could take them. In my classes, both with biology and anatomy and physiology, you can expect kind of a couple of different things. One, I am a very fast grader, and I will give copious amounts of feedback with any assignment that you turn in. My classes largely look the same. Prior to any class, you are going to normally have outside reading that you must complete. That is typically in the form of your textbook, but I typically also provide videos or other materials that you can use to help kind of supplement that material in the textbook. One difference between anatomy and biology is that biology will have what is known as a pre-lecture question prior to coming to class. It is affectionately known as a PLQ, but it's a simple question that they must answer that's related to the reading that they had to do outside of class. Once my students get into class, I am largely lecture-based because of the nature of the virtual world, but then I also try to always include group work into our assignments, into our topics, especially if it is something that needs to have some kind of hands-on practice, like genetics or tonicity or something along those lines. After class is complete, students will typically have maybe a discussion board or a unit quiz that they will have to prep for or answer, but then that largely kind of concludes the outside responsibilities that they would have when we are not meeting. One cool thing that we have for both is we do have fall and spring projects that will be completed. There's going to be one in the fall, one in the spring, and that helps to kind of broaden and also kind of hopefully find that area of science that they really enjoy. Now, in order to let you see a little bit more of me and how I teach, please enjoy my sample teaching class. Welcome to my teaching sample. To give a little bit of background for this little just snippet of my classroom and what I typically do as a teacher. Prior to this, we have covered the cell cycle, which involves interphase, cell division, and then cytokinesis. And so this part of my lecture is going to go into actually talking about the 3 major steps of DNA replication and how the overall process actually occurs. So without further ado, let's begin. Step number 1 of DNA replication is known as unwinding. However, I will often refer to it as unzipping because that's the verbiage that I was taught when I was growing up. So if that happens to come out, I wanted to make sure that you are aware that unzipping and unwinding are the same thing. So the hallmark of the first step, when you unzip or unwind the DNA helix in order to do DNA replication, your enzyme that's doing the work is known as DNA helicase, which makes sense because you have a helix, your DNA helices, that needs to be opened up in order to create a space that allows for step 2 and then step 3 to occur. And so DNA helicase will recognize a specific sequence of DNA. It will attach to the strands. And then as it moves up, what it does is it breaks these hydrogen bonds that exist between your nitrogenous bases, the T, the A, the G, the C. And it will break those hydrogen bonds in order to create an opening known as the replication fork in order for step number 2 and then eventually step number 3 to occur. So the DNA helicase unwinds or unzips the helix of your DNA in order to create this space that allows for the rest of the process to happen. Now, as a side note, before I continue this lecture, whenever I include a slide that has a picture on it, whether I drew it or it's a diagram from the book or something along those lines, and it doesn't seem like it has a lot of things on it, that is because I'm going to go through and I'm going to talk about it, because I firmly believe that students are mostly visual learners. But I always provide a slide, dun, 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 on the next one that will detail the important information that I talk about in that previous slide. And then you have the words to go along with that picture. Going back to our lesson. In step number 2, that is known as base pairing. And this is where all the nitty gritty stuff happens of DNA replication. And this is where you actually start to construct those new daughter strands that are going to eventually create the new DNA molecules. However, there's a little bit of a wrench that gets thrown into this process, because the big enzyme that works here, which is known as DNA polymerase, can only move in one direction, and that is this 3' to 5' direction. Just as a little side note for this, the 3' and 5' refer to the carbons that are in the deoxyribose sugar of your DNA molecule, and it just refers to the orientation. Our DNA molecule has what is called an anti-parallel orientation. So one strand of the DNA goes 3' to 5', the other one goes 5' to 3'. And so they have opposite orientations, anti-parallel. That really dictates how this next step occurs with the DNA polymerase, because DNA polymerase can only move in the 3' to 5' direction. So for one of your strands that we call the leading strand, that's not a problem. We just continue on right behind that DNA helicase, and I match up those nitrogenous bases, the new nucleotides that are just hanging out here. I match them to my strand, and then I can build my strand just continuously right behind that DNA helicase. So I bring in my A, my C, my T, my C, my G, and then I will continue to move right behind that DNA helicase and build my strand as I go. But when you flip over to the other side, that's not how that strand is going to work. And we call this the lagging strand, because it takes just a little bit longer in order to build this strand, because the DNA polymerase actually has to move in the opposite direction of DNA helicase. Because DNA polymerase can only go in the 3' to 5' direction, I actually have to attach right behind the DNA helicase and then move down my DNA molecule. So that causes the lagging strand to be made in fragments that we call the Okazaki fragments. They're about 100 base pairs, 200 base pairs in length, and then you are going to have several of them along that lagging strand that will eventually get pieced together. Another important side note for this part of DNA replication is if you take our cells, which in humans we have 23 pairs of DNA, 23 chromosomes of DNA. So collectively, we have 46 altogether. If you are going through S phase of cell cycle in order to replicate your DNA, you have to replicate all of those. And you don't just start at the top of your DNA, one of your chromosomes and go down it. You'd have to do that 46 different ones. You actually have multiple sites of DNA replication all along your chromosomes. So the DNA replication is not only fast and efficient, but it's not going to obviously take forever to complete. And so along with the Okazaki fragments being made in chunks, you also have other areas of DNA replication that's going to exist all along one chromosome, and all of that eventually has to get spliced together in our very last step, which is your base pairing, which is your ligase. So just like on the previous slide, everything would be detailed out in here that you could go back and you can read and recall. So step 3 is known as the joining step, and this is where your hydrogen bonds are going to be reformed. The enzyme that is going to be responsible for starting this is the DNA ligase. And so it will attach to a lower area where you have your new strand and your old strand, and it will bring these nitrogen bases close enough together that the hydrogen bonds will reform. And then as it moves up the strands of DNA, it is going to seal all of the bits and pieces together so that you have 2 identical daughter cells at the end of the whole entire process. Now, going back to an idea that's known as semi-conservative replication. When you replicate DNA, the daughter cells or the daughter DNA molecules that you are going to create are going to be half old and half new.
One strand is going to be what is called the parent strand, the original strand of DNA from that original molecule. And then you have the new strand, which is known as your daughter strand. And so this is where it gets the name of being a semi-conservative replication process, because half of it is old and half of it is new. Going on to my very last slide, here are the words that obviously I just kind of spoke about, and then it leads us into what I would continue with on the rest of our lecture was, do you think this has something to do with telomeres? So thank you very much for joining my sample, and I can't wait to meet you. Bye for now.